Although our nervous system is often compared to a computer, it is more complicated and versatile. Both depend on electrical activity for the rapid flow of information and processing. However, unlike a computer, portions of the brain rework their electrical connections as new information arrives and learning occurs. Along with the endocrine system, discussed in Chapter 19, the nervous system controls and adjusts the activities of other systems. Both the nervous system and endocrine system require chemical communication with target tissues and organs, and they often work together. The nervous system provides swift but brief responses to stimuli by temporarily changing the activity of other organ systems. The response appears in a few milliseconds, but the effect disappears almost as quickly. In contrast, endocrine system responses develop much more slowly than nervous system responses, but they last much longer, hours, days, or years. This chapter considers the structure and function of nervous tissue. Subsequent chapters build on this foundation as we explore the organization of the brain, spinal cord, higher order functions, and our senses of sight, smell, hearing, balance, touch, and pain. As Figure 13.1 shows, the nervous system consists of all the nervous tissue in the body. The nervous system has two main anatomical subdivisions, central and peripheral. The central nervous system, CNS, is composed of the brain and spinal cord. The CNS processes and coordinates sensory input and motor output. It is also the location of higher functions, including intelligence, memory, learning, and emotion. Early in development, the CNS begins as a mass of nervous tissue organized into a hollow tube. As development continues, the tube's central cavity decreases in size, and the thickness of the tube's walls and the diameter of the enclosed space varies from one region to another. The narrow central cavity of the developing spinal cord is called the central canal. The expanded chambers of the brain, called ventricles, are continuous with the central canal. A clear, watery fluid called cerebrospinal fluid, CSF, fills the central canal and ventricles and surrounds the CNS. The peripheral nervous system, abbreviated PNS, consists of all the peripheral nerves and nervous tissue outside of the central nervous system. The peripheral nervous system provides sensory information to the central nervous system and carries motor commands from the central nervous system to peripheral tissues and systems. The peripheral nervous system has two divisions, afferent and efferent, as you see here in figure 13.2. The afferent division of the peripheral nervous system carries sensory information to the central nervous system. The afferent division begins at receptors that monitor specific characteristics of the environment. There are many forms of receptors. A receptor may be a sensory process, a specialized cell or cluster of cells, or a complex sense organ, such as the eye. The stimulation of a receptor carries information to the central nervous system. The afferent division also delivers information provided by special sense organs, such as the eye and ear. The efferent division of the peripheral nervous system carries motor commands from the central nervous system to muscles and glands. The efferent division begins inside the central nervous system and ends at an effector, a muscle cell, gland cell, or another cell specialized to perform specific functions. Both divisions have somatic and visceral components. The afferent division carries information from somatic sensory receptors that monitor skeletal muscles, joints, and the skin from visceral sensory receptors that monitor other internal structures such as smooth muscle, cardiac muscle, glands, and respiratory and digestive organs. The efferent division consists of the somatic nervous system, abbreviated SNS, which controls skeletal muscle contractions, and the autonomic nervous system, abbreviated ANS, or visceral motor system, which regulates smooth muscle, cardiac muscle, and glandular activity. The activities of the somatic nervous system may be voluntary or involuntary. Voluntary contractions of our skeletal muscles are under conscious control. You exert voluntary control over your arm muscles as you raise a glass of water. Involuntary contractions are not under conscious control. If you accidentally place your hand on a hot stove, you will withdraw it immediately, even before you notice the pain. The activities of the autonomic nervous system are usually outside our awareness or control, such as heartbeat, digestive processes, and our instinctive response to threatening situations. Figure 13.3 summarizes the most important concepts and terms introduced into this chapter. Our discussion of the nervous system begins at the cellular level with the histology of nervous tissue. So, let's check in with what we've learned already. What are the two subdivisions of the nervous system? What are the two subdivisions of the efferent division of the peripheral nervous system? Here, we see listed the major anatomical and functional divisions. These are the central nervous system. In the central nervous system, the brain and spinal cord have control centers that process and integrate sensory information, plan and coordinate responses to stimuli, and control activities of other systems. The peripheral nervous system, efferent and afferent nervous tissue that links the central nervous system with sense organs and other systems. And the autonomic nervous system, which is where components of the central nervous system and the peripheral nervous system that control visceral functions. Shown here is the gross anatomy of the nervous system. First, we see neural cortex, which is a layer of gray matter on the surface of the brain. Then we have the center, a group of neuron cell bodies in the central nervous system sharing a common function. Nucleus, a central nervous system center with distinct anatomical boundaries. Tract, a bundle of axons within the central nervous system sharing a common origin, destination, and function. Column, a group of tracts found within a specific region of the spinal cord. Ganglion, an anatomically distinct collection of sensory or motor neuron cell bodies within the peripheral nervous system. And nerve, a bundle of axons in the peripheral nervous system. Shown here is the histology of the nervous system. First, we have the neuron, the basic functional unit of the nervous system, a highly specialized cell, a nerve cell. Then we have the sensory neuron, a neuron whose axon carries sensory information from the peripheral nervous system towards the central nervous system. And then we have the motor neuron, a neuron whose axon carries motor commands from the central nervous system toward effectors. Then we have neuroglia. These are supporting cells that interact with neurons to regulate the extracellular environment, defend against pathogens, and repair nervous tissue. Gray matter, which is nervous tissue dominated by neuron cell bodies. And then we have white matter, nervous tissue dominated by myelinated axons. Then we look at dendrites, neuronal processes that are specialized to respond to specific stimuli in the extracellular environment. The cell body of a neuron. Myelin, a membranous wrapping produced by neuroglia that coats axons and increases the speed of action potential propagation. Axons coated with myelin are myelinated. Axon, a long, slender, cytoplasmic process of a neuron. Axons conduct nerve impulses called action potentials. Key point. The nervous system contains only two types of cells, neurons and neuroglia. Neurons transfer and process information. Neuroglia isolate the neurons, provide a supporting framework, help maintain the intercellular environment, and act as phagocytes. Shown here are the functional categories of the nervous system. First, we see receptors, a specialized cell, dendrite, or organ that responds to specific stimuli in the extracellular environment and whose stimulation alters the level of activity in a sensory neuron. Then we see effectors, a muscle, gland, or other specialized cell or organ that responds to neural stimulation by altering its activity and producing a specific effect. Then we have reflexes, which is a rapid, stereotyped response, always the same, to a specific stimulus. Other functional terminology, somatic, pertaining to the control of skeletal muscle activity, somatic motor, for example, or sensory information from skeletal muscles, tendons, and joints, example, somatic sensory. Visceral, pertaining to the innervation of visceral organs, such as digestion, circulation, etc., visceral motor, or sensory information from visceral organs, example, visceral sensory. Voluntary, under direct conscious control. Involuntary, which is not under direct conscious control. Subconscious, this is relating to centers in the brain that operate outside a person's conscious awareness. And action potential, sudden, transient changes in the membrane potential that are propagated along the surface of an axon or sarcolemma. Also of note, a typical neuron has a cell body or soma. The cell body is attached to an elongated axon or nerve fiber that ends at one or more axon terminals, and we'll talk about that in a minute. Also, nervous tissue contains approximately 100 billion neuroglia, also termed glial cells, approximately 5 times the number of neurons. Neuroglia are smaller than neurons, and unlike neurons, they retain the ability to divide. So let's have a concept checked. What are the two distinct cell types found within nervous tissue? And which of the two cell types found within nervous tissue has retained the ability to divide? In this figure, the relationship of the 4 parts of a neuron, the functional activities of each part, and the normal direction of action potential conduction are shown. The dendrites, stimulated by environmental changes or the activities of other cells. The cell body, which contains the nucleus, mitochondria, ribosomes, and other organelles and inclusions. Axon conducts nerve impulse, action potential, towards axon terminals. And axon terminals affect another neuron or effector organ, muscle or gland. Key point. The 4 types of neuroglia in the central nervous system are astrocytes, oligodendrocytes, microglia, and ependymal cells. The 2 types of neuroglia in the peripheral nervous system are satellite cells and Schwann cells. The greatest variety of neuroglia is found within the central nervous system. Figure 13.5 compares the functions of the major types of neuroglia in the central nervous system and the peripheral nervous system. Key point. Neuroglia of the central nervous system surround central nervous system neurons and hold them in place, isolate neurons from each other, supply oxygen and nutrients to neurons, destroy pathogens, and remove dead or damaged neurons. The largest and most numerous neuroglia are the astrocytes. They maintain blood-brain barrier, provide structural support, regulate ion, nutrient, and dissolved gas concentrations, absorb and recycle neurotransmitters, form scar tissue after injury. A second type of neuroglia within the central nervous system is the oligodendrocyte. These myelate central nervous system axons provide structural framework. The smallest neuroglia possess slender cytoplasmic processes with many fine branches. They remove cell debris, waste, and pathogens by phagocytosis. Ependymal cells. These cells line ventricles, the brain, and central canal, spinal cord. Assist in producing, circulating, and monitoring cerebrospinal fluid. Key point. Satellite cells and Schwann cells of the peripheral nervous system have functions similar to those of the astrocytes and oligodendrocytes of the central nervous system. Satellite cells surround neuron cell bodies and ganglia, regulate oxygen, carbon dioxide, nutrient, and neurotransmitter levels around neurons in ganglia. Schwann cells surround all axons in the peripheral nervous system, myelate peripheral axons, participate in repair process after injury. Here we are seeing a diagrammatic view of nervous tissue in the spinal cord, showing relationships between neurons and neuroglia. The ependymal cell are the simple cuboidal epithelial cells that line fluid-filled passageways within the brain and spinal cord. The microglia are the phagocytes that move through nervous tissue, removing unwanted substances. The astrocyte are the star-shaped cells with projections that anchor to capillaries. They form the blood-brain barrier, which isolates the central nervous system from the general circulation. And the oligodendrocyte are the cells with sheet-like processes that wrap around axons. Time for a concept check. Specifically, what cells help maintain the blood-brain barrier? What is the name of the membranous coating formed by oligodendrocytes around central nervous system axons? In this figure, we see the ependyma, which is a cellular layer that lines the ventricles of the brain and the central canal of the spinal cord. To the left, we see a light micrograph of the ependymal cells lining the central canal. And to the right, we see an SEM of the ciliated surface of the ependyma from the central canal. We notice the cilia that's on the cell. All peripheral nervous system axons are shielded from contact with interstitial fluids by neuroglia called Schwann cells. Schwann cells ensheath many axons in a layered phospholipid covering known as myelin. An axon wrapped in myelin is said to be myelinated. Myelin improves the conduction speed of an action potential or nerve impulse along the axon. An individual Schwann cell produces myelin by wrapping itself around the axon as diagrammed in the photo. In the first figure, we see that In myelating a peripheral axon, a Schwann cell first encloses a segment of the axon within a groove of its cytoplasm. Then in the second figure, the Schwann cell then rotates around the axon. And in the third figure, as the Schwann cell rotates, the inner membranous layers are compressed and the cytoplasm is forced into more superficial layers. When completed, the myelin sheath consists only of the phospholipid bilayers of the plasma membrane, with the Schwann cell nucleus and cytoplasm at the surface. In the figure on the left is a myelinated axon in the peripheral nervous system. Because each Schwann cell myelinates only about 1 millimeter of an axon, it takes many Schwann cells to myelinate an entire axon. The portion of a myelinated nerve axon between two successive Schwann cells is called an internode. The small gaps that separate internodes are called nodes or nodes of Ranvier. As you see in the cross section of a myelinated axon, the myelin sheath appears as concentric, dense lines around the axon. The figure on the right is of an unmyelinated axon in the peripheral nervous system. A single Schwann cell shields multiple unmyelinated axons within superficial grooves. However, a chain of Schwann cells is still needed to enclose an entire axon. Key point: neurons, the functional units of the nervous system, transmit information from one part of the nervous system to another by electrical impulses. The cell body of a representative neuron contains a large, round nucleus with a prominent nucleolus. The cytoplasm of a neuron is the perikaryon. The cytoskeleton of the perikaryon contains neurofilaments and neurotubules, which are similar to the intermediate filaments and microtubules of other types of cells. Bundles of neurofilaments, called neurofibrils, extend into the dendrites and axons, providing internal support. The perikaryon contains organelles that provide energy and synthesize organic materials. The mitochondria, free and fixed ribosomes, and membranes of the rough endoplasmic reticulum give the perikaryon a coarse, grainy appearance. Mitochondria generate ATP to meet the high energy demands of an active neuron. The ribosomes and the rough endoplasmic reticulum synthesize proteins. Some areas of the perikaryon contain clusters of free ribosomes and rough endoplasmic reticulum. These regions, which stain a dark color, are called Nissl bodies, or chromatophilic substance, after the German neurologist Franz Nissl, who first described them. Nissl bodies give a gray color to areas containing neuronal cell bodies, the gray matter seen in gross dissection of the brain or spinal cord. An axon is a long cytoplasmic process capable of propagating an action potential. In a multipolar neuron, a specialized region, the axon hillock connects the initial segment, the base, of the axon to the cell body. The axoplasm, or cytoplasma of the axon, contains neurofibrils, neurotubules, lysosomes, mitochondria, numerous small vesicles, and various enzymes. An axon may branch along its length, producing side branches called collaterals. The axon and collaterals end in fine terminal extensions called telodendria. The telodendria end in axon terminals, where the neuron contacts another cell. Organelles, nutrients, synthesized molecules, and waste move between the cell body and the axon terminals by a process termed axoplasmic transport. This complex process consumes energy and relies on the neural fibrils of the axon and its branches. Figure 1311 shows the four structural groups of neurons. This classification is based on the placement of the cell body and the number of associated processes. This figure shows an axonic neuron. Axonic neurons have more than two processes, but axons cannot be distinguished from dendrites. Anaxonic neurons are small. In these neurons, it is very difficult to distinguish dendrites from axons. Anaxonic neurons are found only in the central nervous system and in special sense organs, and their functions are poorly understood. This figure shows a bipolar neuron. Bipolar neurons have two processes separated by the cell body. Bipolar neurons have a number of fine dendrites that fuse to form a single dendrite. The cell body lies between this single dendrite and the single axon. Bipolar neurons relay sensory information concerning sight, smell, and hearing. Their axons are not myelinated. This figure shows a pseudo-unipolar neuron. Pseudo-unipolar neurons have a single elongate process with the cell body off to one side. These neurons have continuous dendrites and axons. In these neurons, the initial segment lies where the dendrite converge, and the rest of the process is usually considered an axon. Sensory neurons of the peripheral nervous system are pseudo-unipolar, and their axons may be myelinated. Shown here is a multipolar neuron. Multipolar neurons have more than two processes. There is a single axon and multiple dendrites. Multipolar neurons have several dendrites and a single axon with one or more branches. Multipolar neurons are the most common type of neurons in the central nervous system. An example of a multipolar neuron is a motor neuron that connects the central nervous system to skeletal muscles. Neurons are classified functionally into three categories. 1. Sensory neurons that detect stimuli in the peripheral nervous system and send information to the central nervous system. 2. Motor neurons to carry instructions from the central nervous system to peripheral effectors. And 3. Interneurons in the central nervous system that process sensory information and coordinate motor activity. Forming the afferent division of the peripheral nervous system, sensory neurons deliver information about the external or internal environment to the central nervous system. The axons of sensory neurons, called afferent fibers, extend between a sensory receptor and the spinal cord or brain. Somatic sensory neurons transmit information about the outside world and our position within it. Visceral sensory neurons transmit information about internal conditions and the status of other organ systems. Receptors are either the processes of specialized sensory neurons or cells monitored by sensory neurons. There are three categories of receptors. Enteroreceptors monitor the digestive, respiratory, cardiovascular, urinary, and reproductive systems and provide sensations of deep pressure and pain, as well as taste, another special sense. Exteroceptors provide information about the external environment in the form of touch, temperature, and pressure sensations, and the more complex special senses of sight, smell, and hearing. Proprioceptors monitor the position and movement of skeletal muscles and joints. Somatic sensory neurons carry information from exteroceptors and proprioceptors. Visceral sensory neurons carry information from interoceptors. Forming the efferent division of the nervous system, motor neurons stimulate or modify the activity of a peripheral tissue, organ, or organ system. Axons traveling away from the central nervous system are efferent fibers. The two efferent divisions of the peripheral nervous system, the somatic nervous system and the autonomic nervous system, differ in the way they innervate peripheral effectors. The somatic nervous system consists of all the somatic motor neurons innervating skeletal muscles. The cell bodies of these motor neurons lie inside the central nervous system, and their axons extend to the neuromuscular junctions that control skeletal muscles. Most of the activities of the autonomic nervous system are consciously controlled. The autonomic nervous system, discussed in Chapter 17, consists of all the visceral motor neurons innervating peripheral effectors other than skeletal muscles. There are two groups of visceral motor neurons. One group has cell bodies inside the central nervous system, and the other has cell bodies in peripheral ganglia. The neurons inside the central nervous system control the neurons in the peripheral ganglia, and these neurons in turn control the peripheral effectors. Axons extending from the central nervous system to a ganglion are called preganglionic fibers. Axons connecting the ganglion cells with peripheral effectors are postganglionic fibers. This arrangement clearly distinguishes the autonomic, visceral motor, system from the somatic motor system. We have little conscious control over the activities of the autonomic nervous system. Located between the sensory and motor neurons within the brain and spinal cord, inner neurons analyze sensory input and coordinate motor output. The more complex the response to a stimulus, the greater the number of inner neurons involved. Inner neurons are classified as excitatory, releasing excitatory neurotransmitters, or inhibitory, releasing inhibitory neurotransmitters, based on their effects on the postsynaptic membranes of other neurons. Concept checked. Examinations of a tissue sample shows pseudo-unipolar neurons. Are these more likely to be sensory neurons or motor neurons? Key point. Regeneration of nervous tissue refers to the repair of complete neurons, neuroglia, axons, myelin, or synapses. Regeneration in the central nervous system differs from that in the peripheral nervous system in mechanism, speed of regeneration, and amount of regeneration that occurs. In the peripheral nervous system, Schwann cells play an important role in repairing damaged nerves. In Wallerian degeneration, the axon distal to the injury site, distal stump, deteriorates, and macrophages migrate in and phagocytize the debris. Schwann cells in the injured area do not degenerate. Instead, they divide and form a solid cellular cord that follows the path of the original axon, shown in figure 1313 here. Concept checked. What is Wallerian degeneration and where does it occur? Key point. Excitability is the ability of a plasma membrane to respond to an adequate stimulus and generate an action potential. Plasma membranes of skeletal muscle fibers, cardiac muscle cells, some gland cells, and the axonema of most neurons including all multipolar and pseudo-unipolar neurons, are examples of excitable membranes. Shown in Figure 1314 is the structure of a synapse. A synapse is the site of communication between a neuron and another cell. This figure shows the structure of a typical synapse. An action potential or nerve impulse is the change in membrane potential that develops after the axon lemma is stimulated to a level known as the threshold. Concept checked. Defined excitability. A neuron may innervate 1. Other neurons, 2. Skeletal muscle fibers, or 3. Gland cells. A single neuron would not innervate all 3. Key point. A synapse between neurons involves an axon terminal and 1. A dendrite, axodendritic. 2. Cell body, axosomatic. Or 3. Axon, axonic. At an axon terminal, a nerve impulse triggers events at a synapse that transfers the information to another neuron or cell. A synapse is a site of intracellular communication between a neuron and another cell. A synapse may be chemical, which is vesicular, or electrical, which is non-vesicular. Another key point. Chemical synapses or vesicular synapses are the most abundant type of synapse. These synapses involve the passage of neurotransmitters between cells. At a typical chemical synapse, also termed a vesicular synapse, a neurotransmitter released at the presynaptic membrane of an axon terminal binds to receptor proteins on the postsynaptic membrane. The neurotransmitter then triggers a temporary change in the membrane potential of the postsynaptic cell. Only the presynaptic membrane releases neurotransmitter, so communication occurs in one direction only, from the presynaptic neuron to the postsynaptic neuron, as shown here in Figure 1314b. Neuromuscular junctions are chemical synapses releasing the neurotransmitter acetylcholine. All somatic neuromuscular junctions utilize acetylcholine. It is also released at many chemical synapses in the central nervous system and peripheral nervous system. The general sequence of events is similar regardless of the location of the synapse or the type of neurotransmitter. There may be thousands of chemical synapses on the cell body of a single neuron, as shown in Figure 1314c. Many are active at any given moment, releasing a variety of different neurotransmitters. Some will have excitatory effects, others inhibitory effects. The activity of the receptor neuron depends on the sum of all the excitatory and inhibitory stimuli influencing the axon hillock at any given moment. Key point. Electrical synapses or non-vesicular synapses are relatively rare and are found between neurons in both the central nervous system and peripheral nervous system. At these synapses, the presynaptic and postsynaptic membranes form a gap junction. Key point. Neurons are the basic building blocks of the nervous system. The billions of neurons within the central nervous system are organized into a much smaller number of neuronal pools. A neuronal pool is a group of interconnected neurons with specific functions. Neuronal pools are defined on the basis of function rather than anatomy. They may be diffuse, involving neurons in several different regions of the brain, or localized, with all the neurons restricted to one specific location in the brain or spinal cord. Each neuronal pool has a limited number of input sources and output destinations, and the pool may contain both excitatory and inhibitory neurons. The basic wiring pattern in a neuronal pool is called a neural circuit. A neural circuit has one of the following functions. Divergence is the spread of information from one neuron to several neurons or from one pool to multiple pools, as shown in Figure 1315a. Divergence allows the broad distribution of a specific input, as when sensory neurons bring information into the central nervous system. The information is then distributed to neuronal pools throughout the spinal cord and brain. For example, visual information arriving from the eyes reaches your consciousness at the same time it is distributed to areas of the brain that subconsciously control posture and balance. In convergence, several neurons synapse on the same postsynaptic neuron, as shown in Figure 1315b. Convergence permits the variable control of motor neurons by providing a mechanism for their voluntary and involuntary control. For example, the movements of your diaphragm and ribs are controlled by respiratory centers in the brain that operate outside of your awareness. However, the same motor neurons can also be controlled voluntarily, as when you take a deep breath and hold it. Two different neuronal pools are involved, both synapsing on the same motor neuron. Serial processing relays information in a stepwise sequence from one neuron to another or from one neuronal pool to the next, as shown in Figure 1315c. Serial processing occurs when sensory information is relayed from one processing center in the brain to another. For example, pain sensations on their way to your consciousness make stops at two neuronal pools along the pain pathway. Parallel processing occurs when several neurons or neuronal pools are processing the same information at one time, as shown in Figure 1315d. Thanks to parallel processing, many different responses occur simultaneously. For example, stepping on a sharp object stimulates sensory neurons that distribute the information to a number of neuronal pools. As a result of parallel processing, you might withdraw your foot, shift your weight, move your arms, feel the pain, and shout, "Ouch!" at about the same time. Reverberation uses positive feedback. In this arrangement, collateral axons extend back toward the source of an impulse and further stimulate the presynaptic neurons, as shown in Figure 1315e. Once a reverberating circuit is activated, it will continue to function until synaptic fatigue or inhibitory stimulus breaks the cycle. Reverberation can occur within a single neuronal pool, or it may involve a series of interconnected pools. Highly complicated examples of reverberation among neuronal pools in the brain may help maintain consciousness, muscular coordination, and normal breathing. Concept checked. Distinguish between a neuronal pool whose function is divergence and a neuronal pool whose function is convergence. Key point. The function of the nervous system depends on interactions between neurons in neuronal pools. The most complex neural processing occurs in the central nervous system. Arriving sensory information and outgoing motor commands are carried by the peripheral nervous system. Axons and cell bodies in the central nervous system and peripheral nervous system are not randomly scattered. Instead, they form masses or bundles with distinct anatomical boundaries. Figure 1316, as well as 13.3, summarize the anatomical organization of the nervous system. In the peripheral nervous system, the cell bodies of sensory neurons and visceral motor neurons are found in ganglia. Axons are bundled together in nerves, with spinal nerves connected to the spinal cord and cranial nerves connected to the brain. In the central nervous system, a collection of neuron cell bodies with a common function is called a center. A center with a distinct anatomical boundary is called a nucleus. A layer of gray matter called the neutral cortex covers portions of the brain surface. The term higher center refers to the most complex integration centers, nuclei, and cortical areas of the brain. The white matter of the central nervous system contains bundles of axons that share common origins, destinations, and functions. These bundles are called tracts. Tracts in the spinal cord form larger groups called columns. The centers and pathways that link the brain with the rest of the body are called tracts. For example, ascending or sensory tracts distribute information from peripheral receptors to processing centers in the brain. Descending or motor tracts begin at central nervous system centers concerned with motor control and end at the effectors they control. Concept checked. Describe the following anatomical structures that occur within the central nervous system: center, tract, and pathway.